Hello, dear students. I welcome you all on behalf of Sri Jendra Rangjon classes. Dear student, we are going to start today a new chapter that is plant growth and development. In this chapter, we are going to talk about two important things that is growth and development. Okay, and how these two differentiate and different parameters regarding to this we are going to discuss. So if we talk about the introductory part, then we have to think and we think that what is growth? We ask usually this question as we start this chapter. So if we talk about growth, it can be defined as an irreversible permanent increase in size. What it is? It is obviously a growth. Growth is what? A increase in size, obviously it can be there. But it should be irreversible and it should be permanent increase in size of an organism or its parts or even of an individual cell. That can be called as called growth. Means growth can be observed in an organism or in the parts of the particular plant or in even single cell also. And all plant organs are made up of variety of tissue. We know that cell are going to form a tissue and tissue are going to form organ. Now the question is there any relationship between the structure of a cell, a tissue, an organ, and the function of a plant they form? That is one of the important question. That is there any relationship like that, the structure and the function? Then yes, all cells of a plant are a descendant of the zygote. So because of this, they are derived from a same type of cell. They have a some relationship between the structure and the function they perform. So you can see this is the diagram of a image of a human being growth where you can see the increase in the height, increase in the growth. You can see in plants also growth takes place during the secondary growth and the wood formation takes place. That is also considered as a growth. Plant whole plant develop from the seed germination, and in seed germination also you can see here first a primary root formation take place, then the root will establish, then you will see the development of a cotyledon, then axillary bud will be formed, and ultimately cotyledon will be shriveled and the new whole plant will be derived. This way the whole new plant can develop from a single, you can say, the cell as well. So to begin with, it is essential to know the development of a mature plant from the zygote. And obviously zygote is present in the particularly seed, and seed germination will going to form a whole mature plant. And this development from zygote to whole plant follow precise and highly ordered succession of an event, and all this event will lead to the formation of this particular one. During this process, a complex body organization is formed, whereby root, leaves, branches, flower, fruit, and seeds are produced, and eventually they are going to die. Okay, so this all are going to produce and eventually the particular plant is going to die. And for the formation of zygote also we know that a sperm and egg cell fertilize and form a zygote, and this zygote is going to form a new plant. And obviously zygote is present in the seed and form an embryo and further development take place. So here a seed is obvious it is derived from the ovule, so it is a matured ovary, nothing but. Growth is an increase in organism or its parts, and it is obviously it should be a irreversible permanent increase. So both 1 and 3 should be the correct answer. Now we have to define a growth in a proper way. So as growth we have discussed, it is a irreversible permanent increase in the size of an organ or its parts or even individual cell, and it is regarded as one of the most fundamental and conspicuous, means visible characteristic of living being. If a growth occur in any organism, we can see the growth is happening because the size increase in size can be easily observed. And this growth can be defined as an irreversible permanent increase, as we have discussed. This you can see, this is the growth is happening, the diagram. So growth occur at the expense of energy. Obvious. Growth is going to take place at the expense of energy. ATP is going to be used. And thus growth is accomplished by a metabolic process, that is both anabolic and catabolic. And if anabolism or anabolic process are more than the catabolism, you will see the growth is happening. So let us consider an example of growth. That is the expansion of a leaf in a growth. Okay, expansion of leaf is obviously a growth because the size is going to be increased. So how would you describe the swelling of a piece of wood when placed in water? The important question is how we can describe a particular swelling of piece of wood that is placed in water. So it is an example of imbibition, not a growth, obviously. This particularly example of imbibition, that is not of a growth. So generally plant growth is considered as indeterminate, and it is obviously a unique feature because of the presence of a meristem at a certain location in the plant body. So why plant growth is generally indeterminate? Because plant retain the capacity of unlimited growth throughout the lifespan. And why the plant retain the growth, particularly a capacity of a growth unlimited throughout the lifespan? Because this ability of plant is due to the presence of meristem at certain location in their body. The cells of such meristem have the capacity to divide and self-perpetuate. Thus, the product, however, soon loses the capacity to divide, and such cell make the plant body. So meristem have the capacity to divide and self-perpetuate, obviously. And this form of growth, where a new cells are always being added to the plant body by the activity of meristem, it is obviously called as the open form of growth. So meristem is helping in the forming of this new cells. And again, wherever the meristematic tissue are present, that also it is going to happen. So that what would happen if meristem ceases to divide? Obviously, there should be no growth in the plant. Then the apical meristem of the root and shoot is responsible for the primary growth of the plant and principally contribute for the elongation of plant along their axis. That is shoot apical meristem in the elongation of the shoot and the root apical meristem in the elongation of the root. And such a way the elongation of the plant axis takes place. And you can see this is the diagram of a shoot where you will see the apical meristem of the shoot like this. And this is covered by the developing leaves, which is called as leaf primordia. And this is the diagram of this section. Same way, this is the root tip, which is showing the presence of apical meristem. Apart from this, in dicot, plant and gymnosperm, lateral meristem, vascular cambium, and cork cambium appear later in life. And this are the particularly secondary meristem. These are the meristem that cause the increase in the growth of organ in which they are active, and obviously they are helping in the secondary growth. So this type of growth is known as secondary growth. So obviously apical meristems are helping in the lengthwise growth. So there is a primary meristem, and this secondary meristem like lateral meristem, vascular meristem, and cork meristem helping in later in their life in the particularly girthwise growth, that is the secondary growth. So this is the primary growth, and this sidewise growth is considered as a secondary growth. Okay. Now, so here obviously meristematic tissue are involved. So we can call this as a localized growth. Okay, we can particularly define a plant growth as localized as well. So I'm writing here one point: plant growth is localized. Okay, so plant growth is localized means plant growth is restricted to certain location where the apical meristem, intercalary meristem, and lateral meristem are present within the plant body. Now, after this, considering the type of growth, that is the primary growth, secondary growth, unlimited growth, localized growth, open form of growth in case of the plant, if growth take place, how you will measure it? And to measure the growth at the cellular level, it is difficult. So growth at a cellular level is principally a consequence of the increase in the amount of protoplasm. Obviously, if cell grows in the size, that is due to the what increase in the protoplasm. If cell divides, that is also a result of increase in protoplasm ultimately. So, to growth occur in any organ, any tissue, any organism, the growth in the protoplasm is the one of the important event, and that is the principal event. And because of this, ultimately cellular growth takes place, or tissue level growth takes place. And to measure the increase in the protoplasm is difficult to measure it directly. So, generally measures some quantity which are proportional to, more or less proportional to it. We measure that quantities, and these quantities help us to make the growth measurable or to measure the growth. And growth is generally measured by a variety of parameters. There are different parameters which are more or less proportional to it. So if we talk about some of them parameters, then the first one is the increase in the fresh weight. Dry weight can also be there. Okay. So increase in fresh weight and dry weight. Fresh weight is a weight along with the water, and the dry weight is the weight without water content. So to get a accurate growth result, the dry weight is considered more. Apart from this, the second we can use a increase in length as a one of the another measure, where a growth of pollen tube can be measured by increase in length. The third, increase in surface area we can measure. An increase in surface area can be measured by a growth in the surface area of a leaf. And if you want to measure this, suppose if this particular leaf is there, then we put a on paper and we take a outline. Then after a certain period of growth time, we allow it to take a growth. And if the growth is observed like this, then we can say this much surface area has increased. So by this dotted line on the page, we can measure the increase in the surface area. Then to measure a plant growth, we can use another parameter, that is increase in a volume. And this increase in volume can also help. Another measure is an increase in cell number, and another parameter which is proportional to it, that is size, that is an increase in the size of a cell. Now increase in girth also take considered as one of the particular parameter. Now if you want to talk about the increase in number of cells, then we can take an example like a maize root apical meristem, that can give rise to more than 17,500 new cells per hour. Be remember one of the important point. Root apical meristem gives us 17,500 new cells per hour. So that is a example of increase in cell number. Then another example is a watermelon cell that may increase in size by up to a 3,50,000 times. So it is an example obvious increase in the cell size or the volume. So in the former, growth is expressed as a number of cells obviously, while in the later it is expressed growth in the increase in the size of the cell. So these are the different parameter are used, and the growth of pollen tube is measured in terms of length, as we have discussed. This is the example of growth in length. In the dorsiventral leaf, the growth is measured in terms of the increase in surface area, as we have discussed. So this is the different parameters which are used to make a growth. Now, after this is the what in different ways in which the or different parameters in which the growth can be measured. Now we are going to talk about the methods of growth measurement. And if we talk about the methods of growth measurement, then the first method is by direct observation. Then second method is by oxynometer. And third method is by crescograph, which is the apparatus developed by J.C. Bose. So crescograph is the apparatus which is developed by J.C. Bose, whereas Direct observation can be used and oxynometer can be used. So this is the directly we can measure this. But second one, the oxynometer way is one of the most important way for us to study. Now there are two type of oxynometer we are going to discuss here, that is arc oxynometer and another one is the Pfeffer oxynometer. So if we discuss this, first we are going to discuss the arc or lever oxynometer. So first we are going to talk about the arc or lever oxynometer. We are going to understand this arc or lever oxynometer by a diagram. Okay. So to understand that, suppose this is a platform and we are taking a potted plant and we are going this potted plant and we are tying one thread here and we are passing this thread over one pulley which is like this, and here we attach some weight. Now to this pulley we're measuring tap or one scale is added like this, and it has one pointer like this. Now what happens, and this is completely arranged over the stand. Okay, so this is the stand over which this particular thing is added, and this is the pointer. This is a potato. Now what happens if the growth take place in height in this particular plant? This thread of silk cotton will move upward side, and due to the weight on this side, this is going to downward, and due to this the rotation of this pulley will take place, and when this pulley will rotate, this pointer will shift from its original position to this side. So this shifting in position will help us to identify or measure the actual increase in length. So this arc oxynometer will help us in a particular measuring increase in length, and it will look like this. But one modified one is the Pfeffer oxynometer that we are going to discuss here. So next one is the second type of oxynometer, which is known as somewhat modified one, which is known as Pfeffer oxynometer. Now this Pfeffer oxynometer, if we talk, it is somewhat like this. Here in this two pulley are taken. One is small one and one is the larger one. Now in this pulley are arranged over some stand like this. And in this, what we can do, here over this bigger pulley, one weight is added, and here one potted plant is taken. Over this pulley, one thread is passed over this small pulley and weight is added on this side, and here on the larger pulley weight is added on the both side. Now here one rotating cylinder is kept here, which keeps on rotating like this. So it is a rotating cylinder, and over this rotating cylinder one black ash paper is coated over this, and here one pointer is attached with this thread of this larger pulley. Now what happens, this will make a one circle initially. If suppose this pointer is here, that time what it will happen, it will make a line here. But as the growth will take place, this pulley will, this line thread will go upside and this will come side, this pulley will rotate. Due to the rotation of this small pulley, the larger pulley will rotate and the weight goes downside, so this weight goes upside and this pointer will reach here. So initially it was making a point here, but after this it will make a line here or circle here after growth. Then this which help us to identify the growth actually which is taking place. So this is the what paper axonometer. So these two axonometer help us to identify the accurate increase in the length. Okay. Now next, after this particularly measurement of growth, we are going to study the phases of growth. And if you want to understand the phases of growth, the period of growth is generally divided into three phases. That is the meristematic phase, where the root tip and shoot apex represent the meristematic phase. Obviously, the shoot root tip help in the growth of the root, and the shoot tip help in the growth of the shoot. The second one is the elongation phase, and the cells proximal to the meristematic zone are called elongation zone cells, which help in elongation. And the maturation is the more proximal to the phase of elongation, farther away from the apex. Means, if you want to draw it, suppose this is the region of root. These all are the region of root. Okay. So if we talk about root, in root you will see here the meristematic region. This is here elongation region, and here this is the root hair zone or maturation region. Now this is the nearby to the base, so it is a proximal end, and this is a distal end. So you can see this maturation is proximal to the elongation, elongation is proximal to the meristematic, and such a way the arrangement of this all three zones is observed. Now the cells in the meristematic phase, if we talk, the cell when it is dividing, when it is present with the apex, and when it is a meristematic phase of growth, show the following characteristic. It is going to show the division phase, but along with it, it is show some of the characteristic. And the characteristic that the meristematic phase of growth that consists of constantly dividing cells both at the shoot and root apex, as we have seen, and these cells of this region are characterized by they are rich in protoplasm. They are rich in protoplasm, and they possess a large conspicuous nucleus. They have a large conspicuous means visible nucleus. Then the cells of this meristematic phase are thin and cellulosic with the abundant cellulosic cell wall, thin cell wall which is found with the abundant plasmodesmatal connection. They have too much plasmodesmatal connection. Along with this, if we talk about some more characteristics, the intercellular spaces are absent. The spaces between the cells, that is intercellular spaces, are absent. If present, then it is very small. Okay. So this is the what some of the characteristic of the region of maturation phase. Region of meristematic phase. Then after this, the proximal to the meristematic region is found a elongation phase. And elongation phase show the increased vacuolization. Then they have a cell enlargement, and the new cell wall deposition takes place in this elongation phase. Okay, so this way the cell gets elongation and elongated, and after elongation, these cell are going to be a part of maturation phase or they stop dividing. So cells of phase of maturation show of maximum size in the terms of the wall thickening and protoplasmic modification. Okay, and these cells are proximal to the phase of elongation, and this represents the maturation phase. Here, these cells stop dividing, okay, and they become a part of maturation zone. In maturation zone, in root, you will see the presence of what are different, particularly we can say the root hairs. So if we talk about some MCQs, growth is accomplished by, obviously it is accomplished by both one and two, that is anabolic and a catabolic process. Both are required. What happens if meristematics to divide? Obviously, if the more growth cannot happen, no growth will be observed. Growth will be stopping. Generally, phases of growth is divided into three phases. That is a meristematic, then elongation and maturation zone. By what process felling of piece of wood occur when placed in water? Obviously, we have just understood in the initial part that it is due to the imbibition, not due to the growth. Which of the following is a character of a meristem? Rich in protoplasm, conspicuous nuclei, cell wall are primary, all. Obviously, all are the characteristic of a meristematic cell. Which of the following meristem involved in secondary growth? Apical meristem involved in primary growth and intercalary meristem also involved in primary growth because both are the primary in origin. Whereas lateral meristem are involved in secondary growth. And this lateral meristem obviously include what? A particular vascular cambium and cork cambium. These both are involved in lateral one. So next is the growth rate. Now if we talk about the growth rate, the growth rate, the increased growth per unit time is termed as growth rate. The increased growth per unit time is termed as what? A growth rate. Now increased growth that can be in a particular unit time, then it is defined as a growth rate. Thus the rate of growth can be expressed mathematically. So an organism or a part of organism can produce more cells in two ways. There are the two different ways in these, or more than cells in a variety of ways, and two ways we are going to discuss. So growth rate shows an increase that may be arithmetic or geometric. So these two type of growth we are going to discuss. So first we are going to talk about the arithmetic growth. So this is the two type. First type is of arithmetic growth. What is arithmetic growth? So in arithmetic growth, you will see that growth follows a mitotic cell division, obviously. But if a one cell undergo mitotic division, only one of the daughter cell continue to divide while the other differentiate and mature. So if we talk about diagrammatic representation, suppose this is the two cells. These two cells divide. These are the two cells from the parent cell. Out of this, one stop dividing and one keep on dividing. Again, out of this, the one stop dividing and one keep dividing. See, these are the stopping the division and these are continuing a division. This cell is continuing a division, these cells are stopping a division. Okay, same happens the further they are continuing a division and they are stopping the division. So if each out of two daughter cell, if one stopping a division and others keep on dividing, and one cell keep on dividing, then it is known as arithmetic jump. So the simplest expression of arithmetic growth is exemplified by a root elongation at a constant rate. So what we have understood, suppose this is a cell, out of this two cell are produced, one stop dividing and become mature and differentiate, and one keep on dividing and produce a two cell, then it is arithmetic zone. Now the simple expression of arithmetic zone growth is exemplified by a root elongation at a constant rate. So on plotting the length of the organ against the time, the linear curve is often like this. If we draw a curve, it is of the height of the plant with respect to time, you will see a constant increase in this, that is a linear curve. Then mathematically it is expressed as LT is equal to L0 plus RT. Means in arithmetic growth if we talk, the LT is a where LT is equal to length at the time t, L0 is the length at time 0, and R represent a growth rate or elongation per unit time. So on plotting the length of the root graph, you will see thus a linear curve as we have seen, but the T is the time of growth. Now here we have seen this particular one, that is a what, a particularly a constant rate increase, and we can take an example. Another type of growth is a geometric growth, and in geometric growth the cells undergo mitotic cell division and produce two daughter progeny, and both of the progeny cell following a mitotic cell division retain the ability to divide and continue to do so. So none is going to get mature. So let us take one example: growth curve of a cell in culture. Growth curve of a cell means a particular bacteria or a fungus is going to grow in a culture. Then the increase in their cell number can be represented by geometric growth. So diagrammatic representation is like this. You can see here. Here these two cells are there. Both are dividing and forming a four cells. These four cells are forming a eight cells. Eight cells are forming a 16 cells. 16 cells forming a 32 cells. So here no one is getting stopping its division. So geometric growth can be exemplified here also. Here one cell is dead after division forming a two cell. After two cell it is going to four cell. After four it is eight, and then it is going to 60. So the initial growth is slow, but if you want to make a curve of this, we can make a curve particularly that is a S-shaped curve. So they are explaining the curve, and the curve look like this. Okay, here we take a time, and here we'll take a size or weight of organ. In this is the lag phase, this is log phase, and this is stationary phase. Now what happens here? This lag phase is considered as preparatory phase, where the initial growth of a is slow. Okay. Next to lag phase, you will see the rapid increase in the growth at exponential rate, that is a log or exponential phase. And here in exponential rate, the both the progeny cell following a mitotic cell division retain the ability to divide and continue so on. So the exponential grows 2 to 4, 4 to 8, 8 to 16. Like this, the cell are going to be increased. But after a certain period of time, the nutrient will be limited, and the nutrient supply become limited. So the growth slow down and lead to the stationary phase. Okay. So this way, the particularly growth rate will be what, a stop. So here, if we plot a curve or a parameter of growth, the size and weight in the geometric growth, when we plot the parameter of growth, that is the size and weight against a time, a typical sigmoid or a shaped curve is obtained like this. In this, we draw a curve against the of size and weight against the time. An exponential phase is obtained, stationary phase and lag phase is there. Now here we remember this is the lag phase, this is the exponential phase, and this is the stationary phase. Okay, so like this, these three phases are observed. Sigmoid curve is a characteristic of the living organism growing in a natural environment. If natural environment is there, obviously it should be having a limited nutrient. And in limited nutrient conditions, sigmoid curve is obtained, as stable curve is obtained. Now this type of growth is common in cells, tissue and organ of a plant, the common one. Okay. So the exponential growth can be expressed as W1 is equal to W0 e raised to RT. Mathematically it can be expressed as W1 is equal to W0 e raised to RT, where W1 is equal to final size, that is a weight, height and number, etc. There is a final weight, final height or final number of leaves or cells. Final size at the initial size at the beginning of the period, that is represented by W0. R is a growth rate, T is the time of the growth, and B is the base of natural logarithm. Now here the R is represented as a relative growth rate and is also the measure of the ability of a plant to produce a new plant material referred as efficient use. It is obviously a measurement of the ability of plant to produce a new plant, and that obviously it should be referred by a efficiency index. Hence, final size of a W1 depends upon the initial size, that is W0. Okay. Now here final size of S is depend on the W0. So diagrammatic representation of the stages during the embryo development, if we show, then you will see this diagram which is given in NCERT. Now here we have a zygote, and this zygote is getting divided. So it can divide into two cells. Out of two, the four cells will be produced. Out of four, these four cells further undergo a fertilization and eight cells are going to be produced. So initially you will follow the geometric phase because all cells are dividing. But after a certain period of time, when the differentiation starts, arithmetic phase starts, where some cells are getting dividing and some cells are not dividing. Okay, so some cells are capable of division and some cells are not capable of division. So after a certain period of time during the differentiation, the arithmetic phase continues or starts. Now, quantitative comparison, if we carry out between the growth of a living system, then it can be of two types. Means we can talk about the growth we have discussed, that is the arithmetic and geometric growth, but growth rate are of two types: absolute growth rate and the relative growth rate. Now, absolute growth rate, if we talk, then it is a measurement and comparison of the total growth per unit time. Whatever the total growth happened in a particular unit time, it is called a absolute growth rate. And relative growth rate, that is represented by a growth of the given system per unit time as a percentage of initial size, it is called a relative growth rate. So measurement and comparison of the total growth per unit time is called absolute growth rate, whereas the growth of the given system per unit time as a percentage, and it is with respect to the relative to the initial size, then it is known as the relative growth rate. So relative growth rate is explained by a formula, that is a growth of a given system per unit time upon initial size into 100. So if you want to understand, then we carry out a diagrammatic comparison of the absolute and relative growth rate, and it is an NCRT diagram which is given. In the first leaf, you can see here that this A represents this surface area of this particular leaf, and this A surface area of this leaf represents a 5 centimeter square. But after a certain period of time, growth happens, and the growth reaches to the A dash. So A dash represents the surface area after a growth, that is 10 centimeter square. Whereas in B leaf, the initial surface area was 50 centimeter square, but after a certain period of growth, it is B dash, that is 55 centimeter square. Now, in this, if you talk in both leaf, if you note down, in both leaves, that is A and B, absolute growth rate is 5 centimeter square. Absolute growth rate is 5 centimeter square. Okay, so that is both leaves, that is A and B. The absolute growth rate is 5 centimeter square. But if we talk about the relative growth rate of leaf A, then as according to the formula, growth per unit time is 5 centimeter square, because after growth it reaches to 10 centimeter square. And initial cell size is 5 centimeter square into 100, so it is going to be 100% growth, because the initial surface area was 5 centimeter square, and after a growth it becomes a 10 centimeter square. Obviously, 100% relative growth rate is observed. But relative growth rate of leaf B is the growth rate per unit time. It is a 5 centimeter square, but initial size was 50 centimeter square into 100, so it is going to be a 10% relative growth rate in the leaf B. So relative growth rate in both leaves is different, but absolute growth rate same. So absolute is the actual growth which is happened, and relative growth rate is with respect to the reference, that is a initial size. Now we talk about the conditions of the growth. So when we talk about the conditions for the growth, there is a different conditions are there, like water is there, and water provides a medium, obviously, for the enzymatic activity which are needed for the growth. Apart from this, it is also considered that water is required for a plant cell enlargement, which that requires obviously water. And rigidity of a cell. The cell shape is there, and to maintain a cell shape, rigidity of a cell is required, which is help in the extension of the growth of the plant. Thus, we can say plant growth and further development is intimately linked to the water status of the plant. So, water status of the plant is an important one for the growth to happen. Next one is oxygen. On oxygen, obviously help in releasing metabolic energy by the what aerobic respiration. which is essential for the growth activities. Okay, so oxygen help in releasing metabolic energy by the respiration for the essential for the growth. Third is the nutrient, and in nutrients obviously required for the essential for the particular growth, that is the macro and micro essential nutrients are there which is required by the plant for the synthesis of protoplasm and act as a source of energy. Nutrients macro and micro are essential obviously so the synthesis of protoplasm source of energy. Apart from this, next is the every plant has an optimum temperature range best suited for its growth. So temperature is also another factor which is another condition which is required for the growth. So temperature range best suited for the growth, and any deviation from this range could be detrimental for the survival. Okay. Now, environmental signals, okay, are also there, such as light and gravity also affect the growth of the plant. Okay, and light, as we know, light, it is a stimulus for shoot growth. It is stimulus for a shoot growth, that is positive phototropism. Whereas second is the gravity, and gravity it is stimulus for root growth, and that is explains positive geotropism. Okay, so these are the two things in which this can be explained. So as we have discussed, root show the positive geotropism and should show the negative geotropism. So let's discuss some of the questions. What is the mathematical expression of arithmetic growth? So we have discussed that is LT is equal to L0 plus RT. That is the second one. And in this, the first one is also LT is equal to L0 plus RT. Second one is also that, but sorry, first one is multiplied by, second one is plus. So second is the correct answer. LT is the length after time t, L0 is equal to length, initial length, and R is the growth rate, and T is the time. The elongation of a root will show obviously a linear growth rate according to the arithmetic growth, but it is a constant growth. Sigmoid growth curve is obtained in the geometric growth, obviously, where the food supply is limited. Which of the following provides a medium for the enzymatic activities? If we consider then water provides a medium for the enzymatic activity. Which of the following environmental signal affect the certain stage of the obviously light and gravity both affects as we have discussed just now. Okay, so that is all about the introductory part, the conditions, how growth can be measured, the type of growth, and the growth rate. Now, from the next class, we are going to discuss the differentiation, redifferentiation, redifferentiation, and the further development process. I hope, dear students, you have understood well. Thank you. Thank you.